You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Wesley Wierson. He's the founder and CEO of Leah Labs. The website is leahlabs.com. We're talking about curing cancer in dogs. So Wesley, thank you for coming.
Yeah, thank you for having me. I'm excited to be here and talk about Leah Labs today.
Yeah, I spoke to a veterinarian named Barbara Royal, and she told me that I guess the cancer rate in dogs is, hovering around 50%. Is that anywhere close to accurate?
Yeah. So about 50% of dogs over 10 years old die of cancer, and about 1 in 3 dogs total die of cancer. So really, we all have a dog, or we all, have a family member that has a dog, and the odds are pretty great that that dog is unfortunately going to be afflicted with cancer someday.
That's crazy. I know dogs age faster than people, but is this a recent statistic? Has it been like this for many years, or is this increasing recently?
No, I think it's a recent. The statistic is new. It's just coming out. But yeah, this has been around, since we've kind of had dogs. So really, I think the reason that it's becoming, talked about a little bit more is just because the dog population kind of continues to rise. So we're around 90 million dogs in the U.S. right now, and the amount of money that we spend on our companion animals and our furry friends also continues to rise. So it's up to 72 billion right now in the U.S. alone. So I think a number of factors kind of are really showing people how many dogs that there are in the U.S. And how much is spent, and then that's really kind of bringing to the forefront the amount of cancer that is found in dogs, and also the translational model that dogs are for cancer in humans. Back in the early 2000s, the NCI, National Cancer Institute, and the NIH, National Institute of Health, kind of launched this One Health model, where what we learn from treating and studying cancer in humans can also be translated to dogs, and then vice versa. So really, I think it's just a number of factors that really kind of are driving, to people's interest and people's knowledge, the fact that cancer is really a pressing issue and man's best friend.
Well, again, do you think the rate of cancer in dogs is increasing, or it's just being reported more often nowadays, or is it just because we have more dogs with cancer? But
no, I think it's just being reported a little bit more. As far as I know, I'm no veterinarian, full disclosure, but as far as I know, the rates are about the same. Of course, true-bred dogs continue to kind of rise, and so of course that brings some of their own problems with inbreeding all these dog lines, and kind of we're creating mutant dogs really. So that maybe is affecting the cancer rate, but as far as I'm aware, really, cancer has kind of always been an issue. It's just kind of being more obvious and more talked about now.
So what's the premise of Leah Labs? How are you planning to intervene and help dogs?
Yeah, you bet. So really, Leah Labs was founded for a number of reasons. So in order to tell that story, I kind of have to give a little bit about my background. So I just finished my PhD from Iowa State University in December. And during my time there, I studied gene editing technology development. And so what I found and what we filed a patent for is this method of putting DNA into the genome at specific sites. So I'm sure you've heard of CRISPR, or the listeners here have heard of CRISPR. And so really CRISPR is just a tool, that allows us to cut DNA. However, what happens after you cut the DNA is really what the important part is. And so for the moniker, it's kind of like editing a Word document. And so what I found is that using CRISPR, I developed this technology that allows us to put in new paragraphs into an already built essay, such as the genome. And the new paragraphs we to recode the DNA.
So what is the premise of Leah Labs? How do you want to make an intervention in cancer in dogs?
Yeah, it's a great question. So Leah Labs really was founded to bring cell therapies to dogs. And so, there's been recent, big news on CAR T cell therapy, which is FDA approved now for humans to treat lymphoma and leukemias. And a lot of research is going into treating other types of cancer. And so this really works quite well in human cancers that are relapsed or refractory to general chemotherapeutics. And so Leah Labs was kind of founded to really take that great success and then translate that into to dogs. And so really in humans, it's non-Hodgkin's lymphoma is one of the types of cancer that CAR T cell therapy is able to induce remissions and maybe we could even say cure in some cases. And dogs get the same type of cancer. It's called B-cell lymphoma. Mm-hmm. So really, Leah Labs was founded to be a company to kind of translate some of the success of cell therapy in humans and bring that to dogs. And then, of course, there's a whole conversation to have about the reasons why we may want to cure cancer in dogs. So we can talk about that too.
Well, it seems obvious why you want to cure cancer in dogs. I mean, at least for the dog owners and the dogs themselves. But are there other reasons that haven't been mentioned? Will it be a good proxy for people? I mean, what's the other reason?
Yeah. So really, how I mentioned earlier, how dog is a really nice model for cancer in humans, and so we're able to treat dogs that have cancer, that have this type of cancer, B-cell lymphoma. And if we treat them with CAR T-cell therapy, we can actually study how that works in dogs and then be able to translate that back into humans. And so a really good example of that could be in humans right now, there's a lot of toxicities involved with CAR T-cell therapy. And so the idea here could be that if we use the dog as a model to study how to mitigate those toxicities, or how to entirely avoid those toxicities, we could then, learn how to treat humans for those toxicities. And the way this is done right now, it's not really done very well. So the way current CAR T-cell therapy is developed is using immunocompromised mice. So we have mice that are in the lab, they have no immune systems. You transplant in tumors and you treat them with a CAR T-cell therapy product. And then you either are able to kind of cure that mouse of a transplanted cancer or not. And then you kind of move that along down the line and end up, eventually treating a human with that. However, you're not able to see those toxicities until you get into a human. And so using the dog to kind of bridge that gap, we can not only, cure cancer in dogs and have a really great impact on the world, but then also turn around and amass a lot of clinical data in a very relevant system that then kind of goes back and then informs humans as well.
Well, I've heard, though, that CAR T cell therapy is like $1 million or hundreds of thousands for one person. Mm-hmm. So how would that happen in dogs?
Yeah, so the average price is around $400,000 or so right now. And then when you factor in all of the other ancillary costs to actually treat a human, you get up to $750,000 or so. So yeah, there aren't very many dog owners in the world who would be willing to spend that, though I'm sure that there are a few. However, there's a number of reasons that the CAR T-cell therapy in humans is very expensive. So one of them is the fact that they generate their CAR T-cells with the use of virus. And so every single dose of CAR T-cells that goes into a human right now, it costs about $40,000 to $50,000 just for the dose of virus that's used to engineer those T-cells. And then on top of that, you have to generate a manufacturing plant that is GMP grade for that virus and for the entire process, which costs $50–100 million to build that manufacturing plant. And so all of that has to then be amortized over the life of the product. And not to mention just the fact that this is all autologous therapy right now. So you take cells out from a sick human, you engineer those, and then put them back into that same human. So those are some of the reasons why it costs about $400,000. However, Leah Labs really we're doing a totally different thing. So we use gene editing to reprogram the T cells, number one, and number two, we're generating allogenic CAR T cell therapy. So where humans are being treated with autologous CAR T right now. We're developing allogeneic CAR T. So that's another reason why we're using dogs, or another great thing that using the dog can help us model is how to use allogeneic CAR T cell therapy. So those reasons really kind of drop the price point for us. Right now, we can generate a dose of CAR T cell therapy for a dog for around $1,000. And when we get to market, we're really looking to make that about half or even 10% of that cost right now.
Wait, why would it be so much dramatically different? The mechanism, instead of using viruses, you're using a different mechanism, but why can you do that in dogs and why can I not do that in people?
Right. So there is certainly research going on right now for using gene editing to create CAR T cell therapy in humans. But I guess another point that I failed to mention is the fact that this is all regulated via the FDA in humans, which of course makes the manufacturing process a lot more expensive. And so we're actually regulated via the USDA. And so it really only takes us 2 to 3 years to complete a clinical trial and get to market. Whereas in humans, it takes 5 to 10 years. And so all of those costs really add up to create a much more expensive therapy in humans. And so really, someday, of course, the idea is that allogeneic CAR T cell therapy for humans, even though it's regulated by the FDA, the price point will drop tenfold or even 100-fold. And I really think that Leah Labs is positioned to kind of help influence that, to help democratize cell therapies by the gene editing model that we're developing.
It's too bad if more people kept pet mice and loved them as much as dogs. They would have made tremendous headway by now. We've cured all kinds of stuff in mice. But
yeah, exactly.
Do you know if dogs are a better proxy for drugs and how they act in humans, or are they just as good or bad as mice? Like any sense
it's a much
of that?
better proxy. Yeah, it's a much better proxy. So again, that's why in the early 2000s, this One Health initiative was founded by the NIH and NCI, just because of how dogs and humans share almost everything these days, right? We sleep together, we live together, eat some of the same foods, and we just share our entire lives with dogs. And so also a really big point here is that dogs get spontaneous disease. Just like humans do. So in mice, we have to induce disease to study it, generally speaking. And so dogs actually just get the disease naturally. And that is another reason why they're a really great model.
Wait a minute. Most diseases in mice have to be induced. They don't naturally happen. And yet people think that's a good proxy for what would happen in people?
Yeah, because
it's crazy.
Yeah, exactly. So it's the best that we have. So being able to induce disease in mice to then study it, it's kind of silly that we give animals disease to try to study how to make them better, rather than take naturally occurring disease, study how to make it better. And so again, for CAR T cell therapy in particular, we have immunocompromised mice that are mutants. They have no immune system. They have to be kept in gnotobiotic conditions. And then what we do is we transplant tumors into those mice, human tumors into those mice, let the tumor kind of take over the mouse, and then dose them with the CAR T cell therapy and see how that CAR T cell therapy works in the mouse. And so really that's just a terrible model for this. And so we hope that Leah Labs will be able to kind of flip the script a little bit and use dogs to help study these things.
That just sounds crazy to me. I know, I'm preaching to, it's falling on deaf ears, I know. As you're saying this, think about that. We're using genetically modified, homogenized animal models, i.e. mice and rats. We're inducing disease in them that doesn't normally happen, which should tell you that it's not likely to model what will actually happen in a person. Then they're pretty dissimilar to people. And we're expecting to find the correlation between what happens in mice and in people. That's
exactly.
Kind of crazy.
Yeah, it's the best that we've had for a really long time. But I think that dogs are poised to kind of help us change that model. Dogs are dying today of these natural diseases. Certainly Leah Labs does not intend to give any dogs any disease, right? We just want to take dogs that have disease and try to either cure them. Of course, once we have a product that's approved, we will know that we have a certain success rate, and that will be great. but also, just to use the dog as an experimental kind of ethical model where they're going to be dying, they're on their deathbed. We could give them the potential for life again, and then we can learn from that as well. So really, that's why we're so excited about this model that we're building.
Why not, in order to bolster your efforts, why not talk to certain pharmaceutical companies and say, we're going to be running this in dogs anyway. The data is much more likely to help you with what you do, if it's a company that's working on CAR T therapies. So, we'd like a subsidy from you in order to do this because we're going to share the results with you and allow you to use it too in your, eventual hopeful human trials and maybe offset your costs that way and allow you to do a lot more than you currently could do.
Right. So that's certainly kind of on our, the Leah Labs vision board is to have the leverage to be able to approach those companies. We're pretty early. We just kind of launched our operations in January of this year. So we're looking to get a little bit further along in our first asset development before we do go ahead and approach those companies. But of course, the big CAR T players like Novartis, they have some monkey models, of course, that they use. However, we think that the dog could be even more ethical than a monkey for, just those reasons of intelligence.
Well, because you operate under a different regulatory regime that's a lot quicker, a lot cheaper, a lot easier, you may be able to get things through faster and easier and, and inform what they're doing.
Yeah, exactly. That's exactly what we hope to do someday. And so you can even do this outside of the regulatory paths as well in dogs. So veterinarians are actually allowed to treat animals with anything that they want to, provided that it's pure and safe from bacteria and isn't just going to obviously make the animal sick. Veterinarians are allowed to give experimental treatments to dogs under a veterinary client-patient relationship. And so that's really another strong point for being able to work with pharmaceutical companies someday to help build out their products and then also, inform how we move forward with new products as well.
I mean, the scope of what you could do and what you have to do is like really crazy now that I think about it. But again, you got a lot of advantages. Like if you're going to do a trial, you can control for diet much easier in dogs than you can in people. That's a complaint I've heard from a lot of these clinical trials. Oh, you can't control for diet, which I don't agree. I know you can. But in dogs, it seems like it would be far easier to do that, get the compliance you want. So, I mean, what you're doing can really shed light on a tremendous amount of other areas and, get to results pretty quick, or much quicker than in human trials.
Yeah, yep. No, that's exactly why you're hitting it on the head. Yeah, that's exactly why we're really excited about what we have going on.
Well, why the focus on CAR T? Why not maybe start with, I don't know, diet, for instance, and see how that affects? Or do you believe that won't have an effect on cancer?
Yeah, so I think it'll be much easier for me to answer the why CAR T question. So one of our co-founders is Dr. Saad Kandarian. He treats patients currently at the Mayo Clinic with CAR T cell therapy. And so with his expertise, it was kind of a no-brainer to kind of go after CAR T. And not to mention the fact that, truly when it boils down to it, our co-founders are all gene editors. And we have wanted to use gene editing to change the world. Certainly since I started my PhD, even before then, I worked on talons as an undergraduate in 2011. And so really since then, I've been dreaming about using gene editing. And some of our co-founders have been in the gene editing space, for 30 years. And so CAR T cell therapy was just kind of, the first logical step, I guess, after it was proven in humans to use gene editing to create CAR T cell therapy for, all the reasons that I outlined previously on the dog as a model, but also just to be able to be innovating on the manufacturing platform for CAR T cell therapy. So I said, current FDA approved CAR T is all viral-based delivery strategies. And so, with us developing this non-viral gene editing platform, that's really kind of right in our wheelhouse. And so what we develop and patent in that way also can directly translate into the manufacturing process for human CAR T cells. So really CAR T cells, even though it's, one of the more challenging problems to date in cancer, or at least in cancer treatments, we are really excited to, be entering that space.
Well, that's great. Do you have the background to be able to talk about the mechanisms and how CAR-T works and why?
Yeah, that would not necessarily be my entire focus. Certainly, that's our CAR-T founder. So I'm the gene editor and the kind of the leader of the company here. But just generally speaking, yeah. So I do understand what a CAR-T cell is. So CAR-T cells are genetically engineered T cells. So T cells, of course, are found in all mammals, and they help to fight off foreign invaders. And so what scientists in the 90s found out is that if you create this CAR, the chimeric antigen receptor, by fusing essentially an antibody domain to the T cell receptor, you can then reprogram the specificity of T cells to, instead of recognizing foreign invaders via the T cell receptor complex, to recognize foreign invaders or to recognize whatever you tell them to by re-engineering the antibody domain that you put on the T cell receptor. And so then once the T cell receptor encounters its target antigen, then it's able to turn on the killing response that T cells have natively, and release all the different molecules that are able to then kill cells.
So this all stems from the fact that, unfortunately, cancer is good at evading the immune system, and that's why you have to reprogram some of the immune cells to specifically recognize a particular cancer.
Yeah, that's exactly right. So T cells don't normally recognize cancer as foreign, and so this really just kind of reprograms them and helps them go find and bind to the cancer cells in an antibody specific way. So that's also one of the really great things about CAR T cell therapy in general, is that all you have to do is reprogram a single part of the CAR, the antigen receptor domain, and then you can retarget the cells to target these different shapes that are found on different types of cancer.
What insights have you gotten from how cancer affects dogs, that would be applicable to people? Does it tend to advance in the same way? Are there any insights that you've gotten or you've heard from, the investigators you work with, things that they wouldn't normally know?
I couldn't comment on that per se, no. I mean, the literature for B-cell lymphoma in dogs, has been developed over the last, 40 or so years. And we know pretty well that B-cell lymphoma is a really great model of non-Hodgkin's lymphoma in humans. A lot of the same drivers for non-Hodgkin's lymphoma in humans are also present in dogs. And of course, the phenotype, and it's the same cells that get sick and all of that. So those are really nice. That just proves out the model quite a bit.
What have you seen as like the outside public perception? When you talk about using animals for experimentation, it's, people think all the animals are abused and they're in horrible conditions and they're sacrificed. But since you're doing it to help these animals themselves, do you find that it's a much easier path and there's less resistance by groups to you doing this?
Yeah, so we have not quite attracted the attention of the different animal rights groups. However, I'm almost excited, if you will, to engage those groups, just because of how what we are doing really is this ethical animal experimentation, if you will. So we're not giving any animals disease; we're trying to cure the disease that they already have. I have, however, gotten some questions about using gene editing and whether we're creating GMO dogs, and just that kind of thing. And again, those questions excite me to get, because that's kind of an opportunity for science communication and outreach. And so really, of course, we're not creating genetically modified dogs. We are just engineering cells that go into the dogs, and those cells are helping to cure the dog of cancer. So really, you have to think, when you're using gene editing, of like all the stakeholders that are involved here. And so certainly when you are able to offer something that can help someone who may be anti-GMO, when you're able to offer something that's gene modified that can actually help them, of course, they're much more willing to understand and accept that. So we're not super concerned about, the animal rights groups being too worried with what we're up to.
Well, that's the nice thing, is the animals that are being used in experiments, like you said, they already have disease. They're going to die unless you help them. You're helping them. They're going to get the benefit of the help. I mean, it's like in every way, it's just such a better outcome for them. You're not turning them into other creatures. You're just trying to help them restore their health. So there should be, again, I was naive, but there should be no outcry from animal rights groups. You're actually helping the animals. You're trying to help millions of dogs, billions of dogs. Yeah?
Exactly. Yeah.
That's
So,
great.
I mean, of course, the unfortunate truth with any, new, really innovative and experimental drug is that potentially during your research and development, a dog may succumb to the disease either faster because of what you give them, or potentially what you give them ends up causing them to perish. But those are just kind of, what happens in research and development. And of course, anything negative that happens, we're certainly going to be learning from to then be able to move on and create a better therapy.
Yeah, it's funny. Imagine if a requirement of animal experimentation was that you could not induce illness. You could only work on stuff that naturally was occurring in the animals. I mean, that would, I don't know if it's a good or bad thing. It seems like initially when I say it's a good thing, but that would at least benefit the poor animals that are giving their lives, for new treatments, instead of just being used and slaughtered and getting no benefit.
Yeah, exactly. I think it would be a really nice kind of paradigm switch to try to engineer health rather than engineer disease, and then try to give health to an animal.
Well, I mean, for whatever it's worth, I personally think that it's a great thing that you guys are trying to do. I mean, for many, many reasons. So that's kudos to you for doing that. It's got a lot of great hallmarks, I think, of good stuff, in my opinion. So.
Yeah, great. Well, thank you. We're really excited as well.
So what would be some really happy milestones for you to achieve in the next, couple of years?
Yeah. So what's really exciting about what we're developing is that really in the next couple of years, we can get to market with a new therapeutic for dogs. So currently we're just building out kind of the cell engineering platform before we treat our first animal, and all the quality control that needs to happen before we actually are able to put a therapy into a dog. But once we're actually able to then get our therapy into a dog, the USDA path of approval is quite straightforward. So in the next 2 years, we will have treated our first dogs in an experimental setting, and then also treated client-owned animals that are actually sick. So we're really excited about that. And then, we'll be moving through the USDA clinical trial process, where we just have to treat 30 to 50 dogs, show that we are able to induce a clinical response that is greater than the standard of care, that is greater than placebo. And the great thing is there's new regulation with the USDA where we actually don't even have to treat dogs with placebo. So really all we have to do is beat a historical control placebo, which is anywhere from 30 to 50 days of life after a diagnosis for B-cell lymphoma. So really no dogs have to die even in a placebo treatment like this, which is really awesome for our animal rights hope moving forward. And then at the end of year 3, we should be able to be conditionally approved by the USDA and ready to sell our product across the nation.
What about total cost, for a clinical trial for people, all the animals used, and et cetera? I've heard it costs hundreds of millions or billions. What's the price comparison here for dogs?
Right. So we're really looking at only about $5 million to get us through all of R&D and all of the clinical trials needed to get this done, which is 100x to 1,000x cheaper than what can be done in humans for developing new therapeutics. So that's really exciting for us. And then the entering the market and scaling maybe a few more million, but really $5 million in R&D and regulatory costs is all it takes to get us to market.
Okay, well, very good. So what's the best way for folks to get in touch and find out more? Yeah,
so just www.lealabs.com, as you mentioned in the intro, and then you can find me at Wes, W-E-S, at lealabs.com. And we'd be really excited to talk to anyone who wants to know more about what we have going on, and anyone who thinks that we could work together.
That's great. Wes, I'm glad I met you, podcasted with you, and I appreciate you being here.
Yeah, thank you for the time. That was a lot of fun.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.